Good day, student. Today our lecture about general characteristic of gram-positive cocci. Cocci is a rounded microbe that can cause inflammatory disease in humans, which is accompanied by the formation of pus, so are called pyogenic. Pathogenic cocci by sensitivity to gram staining technique classified by two groups. Gram-positive cocci will have violet or blue color, and gram-negative cocci will have a red or pink color. Gram-positive cocci include two families, family Micrococcaceae and family Streptococcaceae. Family Micrococcaceae includes genus Staphylococcus, genus Micrococcus, and genus Stomatococcus. Family Streptococcaceae includes a lot of genus, but we will study in our academic years genus of Streptococcus, because this microorganism very often cause inflammatory disease. Gram-negative cocci include two families, family Neisseria and family Veillonelliaceae. Family Neisseria includes genus Neisseria and another genus, but we will study genus Neisseria in our laboratory class. And family Veillonelliaceae, genus Veillonella. Taxonomy. Staphylococci are included in the Firmicutes bacteria. Family Micrococcaceae or Staphylococcaceae, genus Staphylococcus. Staphylococci are subdivided into more than 13 species, but we will study Staphylococci aureus, Staphylococci epidermidis, and Staphylococci saprophyticus. Species of Staphylococci are initially differentiated by the coagulase test and are classified into two groups, coagulase positive and coagulase negative. Coagulase positive Staphylococci of the 20 species found in man. Staphylococci aureus, formerly also called Staphylococci pyogenes, is coagulase positive. Coagulase negative Staphylococci, there are 19 coagulase negative Staphylococci. Staphylococci epidermidis, Staphylococci saprophyticus are the most clinically significant species in this group. Morphological properties. Staphylococci are spherical in shape, 0.8 to 1 micrometer in diameter, and form irregular clusters resembling bunches of grapes. They may also be found singly, in pairs, and in short chains of 3 or 4 cells, especially when examined from liquid culture. Long chains never occur. They are non-sporing, non-motile, and usually non-capsulate, with the expression of rare strains. On this slide you can see smear Staphylococci aureus. You see gram-positive spherical shaped microorganism and arrangement in the smear clusters like grapes. Cultural properties. Staphylococci are facultative anaerobes by type of respiration, catalase positive, distinguished features from Streptococci, which are catalase negative. They grow well on ordinary nutrient media with pH of 7.2 to 7.4 at temperature of 37 degrees, but do not grow at temperature below 10 and above 45 degrees. Most strains produce golden yellow aurous pigment, though some strains may form white, non-pigmented colonies. On mid-peptone agar, Staphylococci produce well-defined colonies with smooth edges measuring from 1 to 2 to 2.5 mm in diameter. Growth on staphylococci in mid-peptone broth produce diffuse opacity throughout the medium and subsequently a precipitate. In some cases, when there is sufficient aeration, the microorganism form a pellicle on surface of the broth. Staphylococci grows well on coagulated serum. After 24 to 28 hours of incubation, there is usually abundant growth along the inculation swab in liquefaction of gelatin media. On the fourth or fifth day, the gelatin medium resembles funnel filled with fluid. On blood agar, pathogenic staphylococci cause hemolysis of the erythrocytes. On this slide, you can see growth of staphylococci aureus on surface nutrient media with a line salt agar, and you see yellow colony, white colony, and around white colony you can see specific zone lecithinase activity. On this slide you can see how grows staphylococci on blood agar. Around colony of microorganism you can see beta hemolysis. Mannitol salt agar is broth, a selective differential nutrient media for cultivation of staphylococci. It is used to differentiate pathogenic Staphylococcus species from non-pathogenic members of the genus Micrococcus. Staphylococci have positive reaction because acid and product turn the phenol red pH indicator from red to yellow. Negative colony, colorless colony, will grow Micrococcus. On surface Bart Parker agar, you can study three specific features of staphylococci. Colony of staphylococci black, and around colony you see two type of zone. Around colony you see clear zone, it's lecithinase activities, and around zone lecithinase activity you see opaque zone of precipitation, and you will understand about lipase activity of staphylococci. On this slide, this same nutrient media and you see black colony. Around black colony you see lecithinase activity, and around lecithinase zone you see zone lipase activity. Staphylococci aureus growing on DNase agar. Note there is breakdown of DNA in the agar, and there is clear zone. You see this colony of microorganism, it's clear zone around the bacterial growth. Staphylococci epidermidis grows on DNase agar different because around colony of Staphylococci epidermidis you don't see zone DNase activity. Catalase test. Catalase test positive if you will see bubble, and catalase negative test you don't see bubble. Characteristics of Staphylococcus aureus strains. Beta hemolysis, produce clear hemolysis on blood agar. Golden yellow pigment. Coagulase positive test. Greater biochemical activity, ferment mannite. On surface mannitol salt agar you will understand about it. Liquefied gelatin produce phosphatase. Black colonies on potassium telluride blood agar or Baker-Parker agar produce black colonies in the medium contain telluride salt. Produce thermostable nuclease DNase activity. which can be demonstrated by the ability of boiled culture to degrade DNA in a agar diffusion test. Coagulase test. You understand. Staphylococci classified by two groups. Coagulase positive means Staphylococci aureus and coagulase negative means Staphylococci epidermidis, Staphylococci hemolyticus and Staphylococci saprophyticus. You can use two groups method for study coagulase activity. You can use light method and tube method. If you will see, for example, flakes, it's positive test. If you use tube method, if you see clot, it's positive reaction. If you see liquid nutrient media, it's negative reaction. On this slide you can see how grows Staphylococci aureus on surface mid-peptone agar, colony smooth, round and produce yellow pigment. Characteristics: 3 species of genus Staphylococci. Different features between the strains of Staphylococci: its growth, anaerobic growth and fermentation of glucose, fermentation of mannitol, produce coagulase, DNase, phosphatase, produce alpha toxin, protein A in cell wall, and sensitivity to antibiotics novabiocin. Antigenic structure. Polysaccharide A was extracted from pathogenic strains isolated from patients with septicemia, furunculosis, osteomyelitis, and acute conjunctivitis, etc. Polysaccharide B is found in avirulent or non-pathogenic strains. Polysaccharide A and B differ not only in their serological reaction, but also in their chemical structures. Antigen C containing a specific polysaccharide has been recently isolated. Virulence factors or pathogenicity of genus Staphylococci. Factors of adhesion: adhesive proteins, the proteins of outer membrane, outer polysaccharides. Factors of invasion: plasma coagulase, hyaluronidase, fibrinolysine, lecithinase, and another ferment. Factors of aggression: capsule, component of cell wall, protein A, catalase, beta-lactamase. And factors toxogenicity or toxin formation. Microorganism, gram-positive microorganism produce just exotoxin, and Staphylococcus produce membranotoxins, exfoliating toxin, toxic shock syndrome toxin, leukocidin, and different type of enterotoxins. On this slide you can see virulent factors of Staphylococci aureus. I will not repeat these factors. Capsule: polysaccharide surrounding the cell wall, inhibition of personalization, and protect of microorganism against phagocytosis. These main factors of aggression: peptidoglycan activates complements and induces release of inflammatory cytokines. Teichoic acid, an antigenic component of the cell wall, facilitates adhesions. Protein A is the surface protein of Staphylococci aureus which binds immunoglobulin G molecules by the Fc region. In serum, bacteria will bind immunoglobulin G molecules the wrong way around by this non-immune mechanism. In principle, this will disrupt opsonization and phagocytosis. Indeed, mutants of Staphylococcus aureus lacking protein A are more efficiently phagocytosis in vitro and slides with mutants in infection models suggest that protein A enhances virulence of Staphylococci means. Membrane damaging toxin. Alpha toxin, the best characterized by and most potent membrane damaging toxin of Staphylococcus aureus is alpha toxin. Susceptible cells have specific receptors for alpha toxin, which always low concentration of toxin to bind, causing small pores through which monovalent cations can pass. At a high concentration, the toxin reacts non-specifically with membrane lipids, causing larger pores through which divalent cation and small molecules can pass. In human, plates and monocytes are particularly sensitive to alpha toxin. Beta toxin is the sphingomyelinase which damage membranes rich in the lipid. The classical test for beta toxin is lysis of sheep erythrocytes. The majority of human isolated of Staphylococcus aureus do not express beta toxin. Delta hemolysins. The toxin has a wide spectrum of cytolytic activity. It lyses red blood cells of sheep, rabbit, horse, and man. It also possesses lethal dermonecrotic and leukocidal activity. Leukocidin, called the Patton Valentine toxin after its discoverers. Staphylococci aureus can express a alpha toxin that specifically acts on polymorphonuclear leukocytes. Phagocytosis is an important defense against Staphylococci infection, so leukocidin should be a virulence factor. Epidermolytic or exfoliative toxin, shortly AET. This toxin causes scarlet skin syndrome in neonates with widespread blistering and loss of the epidermis and have protease activity. It is not clear how the latter cause epidermal slitting. It is possible that the toxin target is a very specific protein which is involved in maintaining the integrity of epidermis. Enterotoxin, serologically distinct to Staphylococci enterotoxin A2E, G2I, and three subtypes of enterotoxin have been identified. Enterotoxin A is most commonly associated with disease. Enterotoxin C and D are found in contaminated milk product and enterotoxin. These toxins are superantigens, however, capable of including non-specific activation of T cells and cytokine release. The toxin also inhibits pyrogenic, mitogenic, hypotensive, thrombocytopenic, and cytotoxic effect upon organism. Next toxin is toxin shock syndrome toxin 1, TSST1. Staphylococcus aureus is isolated with toxic shock syndrome, are severe and often fatal disorders characterized by multiple organ dysfunction. The enterotoxin and toxic shock syndrome toxin 1 belong to a class of polypeptide known as superantigens, which are potent activator of T lymphocytes, leading to release of cytokines such as interleukins and tumor necrotic factors TNF. This results in clinical conditions of TSS. The systemic effect of this disease results from these process. Extracellular enzymes of Staphylococci: hyaluronidase, lipase and hydrolase, hemolysins and glycosidins, clumping factors A, coagulase, desoxyribonuclease or DNase or nuclease, catalase and penicillinase or beta-lactamase. I will not read full material. You can read in after my lecture in PDF file. Fibrinolysins or staphylokinase. Many strains of Staphylococci aureus express a plasminogen activator called staphylokinase, a complex formed between staphylokinase and plasminogen activates plasmin, like proteolytic activity, which causes dissolution of fibrin clots. The mechanism is identical to streptokinase, which is used in medicine to treat patients who are suffering from coronary thrombosis. As with coagulase, there is no evidence for staphylokinase as a virulence factor. Also, it seems reasonable to imagine that localized fibrinolysin might aid in bacterial spreading. On this slide, you can see major virulence factors of Staphylococci aureus and their biological action: capsule protein A, lipoteichoic and teichoic acid, different type of enzymes, catalase, coagulase, degradative enzymes, penicillinase, and different toxins which produce Staphylococci, example leukocidin, cytolytic toxin, enterotoxin, exfoliative toxin, and toxin shock syndrome toxin. On this slide you can see different characteristics of hemolytic properties of Staphylococci on surface blood agar. And you can see different sensitivity of different species of Staphylococci to novabiocin. Staphylococci aureus around disc with antibiotics novabiocin don't grow. You see clear zone. You have to understand about that Staphylococci aureus sensitive to novabiocin. Staphylococci saprophyticus grows around of disc with antibiotics, and you know that this microorganism in Staphylococci saprophyticus don't sensitive to novabiocin. Epidemiology: source of infection: sick person, bacterial carrier, and food. Staphylococci colonized approximately 13% of general population. Major cause of community-acquired and nosocomial infection. Staphylococci epidermidis, most commonly identified Staphylococcus species from human skin. Common cause of nosocomial bacteremia. Staphylosaprophyticus, common cause of urinary tract infection in sexually activity young women, because have a specific interaction between specific receptors on surface reproduction tract. Pathogenesis and disease: skin and soft tissue infection, folliculitis, impetigo, cellulitis, abscess, staphylococcal scarlet skin syndromes, necrotizing fasciitis, surgical wound infection, bloodstream infection, infective endocarditis, pneumonia, and another disease can cause staphylococci, and food poisoning and urinary tract infection. On this slide you can see which type of toxin cause disease. Toxin mediate disease: food poisoning, because microorganisms produce enterotoxin. Scarlet skin syndrome due to exfoliative toxin. Toxic shock syndrome due to toxic shock syndrome toxic 1 superantigen, and toxic shock syndrome syndrome due to toxic shock syndrome toxin 1, and superactive infection MRCA, carbuncles, endocarditis, impetigo, osteomyelitis and septic arthritis, pneumonia and wound infection can cause Staphylococci. Laboratory diagnosis. Test material may be obtained from pus, mucous membrane discharge, sputum, urine, blood, foodstuff, vomit, lavage fluid, and feces. The material is examined for the presence of pathogenic Staphylococci. Special rules are observed when collecting the material since non-pathogenic strains are widespread in nature. On this slide you can see laboratory algorithms for study gram-positive cocci. Means if you see under microscope cocci spherical shape of microorganism gram-positive, you will take catalase test. If catalase test negative, you will study like Streptococcus genus. If catalase test positive, you will use coagulase test and Nova-Biacin test for understanding about strains of Staphylococci, aureus, epidermidis or saprophyticus. Treatment. Staphylococcal disease are treated with antibiotics, sulfonylamides and anti-staphylococcal gamma globulin. Prophylaxis. The general precautionary measure includes hygiene in working and everyday life conditioning, treatment of vitamin deficiency, prevention of transmits and express perspiration, observe of rules of hygiene in machinery, hospital, surgical department, and another principle of personal hygiene. Road disinfection of hospital premises and bacteriological examination. To prevent pyodermia, protective ointment and plastic are used in industrial enterprises. Next, gram-positive cocci is Streptococcus. Streptococcus family Streptococcaceae, taxonomic position. Family Streptococcaceae, genus Streptococcus includes genus Enterococcus, Aerococcus, and another genus. Species which we will study is Streptococcus pyogenes and Streptococcus pneumonia. Streptococci are identified as different species of blood agar on the basis of their hemolytic pattern and sensitivity to optachin and bacitracins, its antibiotics. There are many pathogenic species of Streptococci, including Streptococci pyrogens, Streptococci agalactica, Streptococci pneumonia, and the virulent Streptococci mutans, mitis, sanguinis. Streptococcal infection causes a wide array of clinical manifestations, including pharyngitis, pneumonia, skin and soft tissue infection, endocarditis, septicemia, and another disease. They are also responsible for the post-infection syndromes of acute rheumatic fever and post-streptococcal glomerulonephritis. Most streptococci are sensitive to penicillin. Morphological properties. Gram-positive cocci, some may lose positive staining after overnight incubation, or if the cultural yeast and the bacteria die. And smear pairs or chains, classic long chains, are best seen when grows in liquid medium. Streptococci non-motile, non-spore forming, facultative anaerobe by type of respiration, maybe obligate anaerobe are distinguished just peptostreptococci. Catalase negative. Catalase is the enzyme that splits hydrogen peroxide into water and oxygen. On this slide you can see chain of Streptococci pyogenes, a smear of pus. Classification. Three different schemes are used to classify the organism as follows. First, hemolytic patterns. Complete beta hemolysins, alpha and gamma hemolysins. Second type classification, serological properties Lancefield groupings. A to H, K to M and O to V antigens. Groups based on serological identification of group-specific C carbohydrates on cell wall. And biochemical classification, physiological, we will study physiological properties and biochemical activity of Streptococci. By hemolytic activity, Streptococci classified by three groups: alpha hemolysis, green zone hemolysis, beta hemolysis, clear zone, and gamma hemolytic without zonal hemolysis. Alpha hemolytic Streptococci includes Streptococcus pneumonia and Streptococcus viridans, means mutans sanguis. Beta hemolytic groups of Streptococci includes Pyrogenes and Agalactia Streptococci, and alpha hemolytics is Enterococcus genus. On this slide you can see hemolytic reaction. If you see clear zone, it's zone beta zone hemolysis. If you see, for example, green zone hemolysis, it's alpha hemolysis. If you don't see zone of hemolysis, it's gamma hemolysis. And different properties between different type of Streptococci. After study hemolytic properties, you will use optachin test and bacitracin test. For example, for understanding different properties between Streptococcus pneumonia, Streptococcus viridans, which give alpha zone hemolysis, you will use optachin test. Streptococci pneumonia will sensitive to optachin. Viridans Streptococci will grow around disc with optachins, optachin resistance. If you will see beta zone hemolysis, you will use bacitracin test. If bacitracin test positive and microorganism grows on surface nutrient media, Streptococcus pyogenes grows around disc with bacitracin, it's Streptococci agalactia. If microorganism don't grow around surface around disc with bacitracin, you have to understand this Streptococcus pyogenes. Cultural properties. Streptococci are facultatively anaerobic, facultative anaerobic, and they are also anaerobic species. The optimal temperature for growth is 37 degrees and no growth occurs below the limits of 20 to 14 degrees. The organism show poor growth on ordinary meat peptone agar and grow well on sugar agar, blood agar, serum agar, and acetic agar and broth. When the pH of the media 7.2 7.6. On solid media they produce small translucent gray or grayish-white colony, and granular colonies with poorly defined margins. On sugar broth, medium growth is in form of fine granular precipitates on the walls and at the bottom of the tube. In only rarely does not broth become turbid. On this slide you can see different type of colony and different zone of hemolysis around colony of microorganism. Biochemical reaction. Streptococci pyrogenesis characterized catalase negative, insoluble in 10% bile unlike Streptococcus pneumonia. It ferments several sugar producing acid and no gas. Fermentation of the sugar has been the basis of physiological classification of the genus, but in the case of hemolytic streptococci, this has been replaced by serological grouping. Hydrolysis of pyrrolidonyl naphthylamine test, or shortly PIR test, is positive and fluor to ferment ribose distinguished it from non-group A hemolytic streptococci. Biochemical reaction you see lysis in bile, which gives Streptococcus mitis, and lysed by bile Streptococcus pneumonia. Different growth, different characteristic of test tube with bile. Identification of facultative gram-positive cocci. First, hemolysis activity. If you see alpha hemolysis activity, you will use bile test and test to optahens. And if you see beta zone hemolysis, you will use test with bacitracin for understanding about different strains of streptococci. Identification. Colonies of Streptococci pyogenes on blood agar are small, transparent, and smooth with well-defined area of beta hemolysis. Gram stain will reveal gram-positive cocci with some short strains. Hemolytic streptococci are grounded by the Lancefield technique using serological method, or biochemical test can be performed. The fluorescence antibody technique has been employed for the rapid identification of group A streptococci. A key test that should be done is bacitracin susceptibility or PIR hydrolysis. Factors pathogenicity, factors of adhesion, Fimbria proteins M, factors of invasion, different ferment, hyaluronidase, fibrinolysin, streptokinase, and another ferment. Factors of adhesion, aggression, capsule, proteins, aminopeptidase, C5a peptidase, crosswise reacting antigens, and toxogenicity factors or toxin formation, hemolysins, erythrogenic toxin, and cardiogepatotoxin. On this slide, you see major virulence factors of group A streptococci. I will not read this slide. You will study at home and our laboratory class. On this slide, you can see virulence factors of streptococci means structure components M protein, lipoteichoic acid, F protein for cell adhesions, hyaluronic acid capsule, enzyme streptokinase, deoxynuclease, C5a peptidase, pyrogenic toxin, and streptolysins, two type, streptolysins O and streptolysin S. This classification streptolysin based by sensitivity to temperature. On this slide, the same slide for your understanding about virulence factors of streptococci and about antigenic structure. Cell wall of streptococci pyogenes consists of inner layer of peptidoglycan, middle layer of group-specific carbohydrate, and the outer layer of protein and lipoteichoic acid containing proteins antigen MTR, type-specific proteins. M proteins act as virulence factors by inhibiting phagocytosis. The capsule of Streptococcus pyogenes composed of hyaluronic acid. Streptococcus pneumonia is capsular polysaccharide. Toxins, hemolysins, alpha, beta, gamma, I will not repeat, and streptolysins O and streptolysin S. Streptolysin O is so called because it is oxygen-labeled hemolysins. Its heat-labeled proteins, cytolytic and capable of lysis erythrocytes, leukocytes, and streptolysins O is strongly antigenic and anti-streptolysins O antibodies appears in sera following streptococci infection. Streptolysin S is oxygen stable and causes hemolysis around the colonies on aerobic blood culture. It is proteins but non-antigenic. It is cell-bound hemolysin that can lyse erythrocytes, leukocytes, and platelets. Pyrogenic exotoxin, erythrogenic scarlatin toxin. The primary effect of the toxin is induction of fever, and so it was remained streptococcal pyrogenic exotoxin. This toxin was originally called erythrogenic. Three immunological distinct type heat-labeled toxin have been described in streptococci pyrogenic. This toxin as super antigen like staphylococci enterotoxin and toxin shock syndrome toxin Y. First, interacting with both macrophage and helper T lymphocyte cells. This causes important effects including fever, shock organ failure, and the rash observed in patients with scarlet fever. Enzymes: desoxyribonuclease, streptodonase or DNase, streptokinase or fibrinolysins, hyaluronidase aids, spreading factors, proteinase, and serum opacity factors. It's lipoproteinase. I will not read full text. Epidemiology. Group A streptococci commonly colonize the oropharynx of healthy children and young adults. There is asymptomatic colonization in upper respiratory tract and transient colonization of skin. The most common route of entry of streptococci pyogenes is the upper respiratory tract. Person to person spread by respiratory droplet or through break in skin after direct contact with infected person. Different disease which cause streptococci, erysipelas, impetigo, pharyngitis, pyoderma, and necrotizing fasciitis. It's infection which can cause streptococci. Toxin-mediated disease, scarlet fever, streptococcal toxic shock syndrome, can cause streptococci, also can cause autoimmune infection. Acute post-streptococcal infection means acute glomerulonephritis and rheumatic fever. Penetrating deep into the tissue, they produce local pyogenic inflammations such as streptodermia, abscesses, phlegmons, lymphadenitis, pharyngitis, cystitis, pyelitis, cholecystitis, and peritonitis, and another infection, and can cause very often without treatment patient can have septic condition because streptococci spread through organism and another systems human body. Laboratory diagnosis. Test material is obtained from the pus of wound, inflammatory exudate, tonsillar swabs, blood, urine, and foodstuff. Procedure are same for staphylococci infection. Test includes microscopy of pus smear, inoculation of the test material into blood agar plates, isolation of the pure culture, and its identification. For treatment of this streptococcal infection, we will use antibiotics and sulfonylamides. Next, microorganism which cause infectional disease, inflammatory disease, Streptococcus pneumonia. In 1881 years, the organism then known as pneumococcus for its role as an etiological agent of pneumonia was first isolated simultaneously and independently by the U.S. Army physician George Stremberg and French chemist Louis Pasteur. Streptococcus pneumonia. Characteristics. Taxonomy. Family Streptococcaceae. Genus Streptococcus. Species Streptococcus pneumonia. It's gram-positive bacteria. 19 serotypes known. Polysaccharide capsule. Important virulent factors. Anti-specific antibody is protective. Morphological properties. Encapsulated, spherical, ovoid, lancer-shaped cocci. Orientation or arrangement, pairs or chains, very often pairs. Gram stain positive. Old culture may be gram negative. Streptococci pneumonia is normally present in the throat and nasopharynx of human. Identification by hemolytic properties. Sensitivity to optachin, disc test. No lens-filled antigens. Positive quelling reaction. Capsular swelling in the presence of specific anti-capsular antibodies. On this slide you can see diplococci, spherical shape, gram-positive color. Similar to other gram-positive organisms, I repeat, lipid bilayer surrounded by region cell wall composed of peptidoglycan and teichoic acid, which contain the determinant for C polysaccharide antigenic activity. Teichoic acid contains amino alcohol choline, regulatory ligand. Plasma membrane contains chlorine containing teichoic acid, which carries the F antigens. Cultural characteristic facultative anaerobes. Cultural media brine hut infusion. Enrichment with 5% defibrinated blood agar. Young cultures of encapsulated pneumococci produce circular, glistening, dome-shaped colonies in 1 mm in diameter. Later, center of colony collapse. Catalase negative, aerobic incubation produce zone alpha hemolysis, and anaerobic incubation, if you use, produce zone of beta hemolysis. This microorganism catalase negative, these different features between streptococci and staphylococci. On this slide you can see colony of microorganism with green zone of hemolysis on surface blood agar. Laboratory identification, you will know about if you see alpha zone hemolysis, you will use optachin test. If you see, for example, clear zone around optagen test is streptococci pneumonia. If you don't see clear zones, it's optagen test negative. Laboratory identification, I repeat, between streptococci pneumonia lysed by bile, streptococci mitis do not lysed by bile. Laboratory identification, covalent reaction, capsular precipitation. Most rapid and most useful identify a specific type of pneumococci in sputum, spinal fluid, exudate or cultural. Pneumococcal specimen mixed with polyvalent anti-pneumococcal serum and methylene blue. And study under microscope. Positive result, refractile and swollen capsule on oil immersion. These pictures before additional of antiserum. This after additional of antiserum swollen capsule. You see different size capsule around pairs of streptococci pneumonia. On this slide you can see another pictures, same reaction. Negative, you see small size of capsule. On this slide you can see large capsule around bacteria. These pneumococcal cells, and around cells you see capsule. Laboratory identification. Animal inoculation, fatal infection within 16-48 hours to mice infected intraperitoneally with sputum infected with pneumococci for experimental purpose. Antigenic properties. Antigenic properties of Streptococcus pneumonia include somatic antigen, C polysaccharide species specific, major cell wall structure components, and 4 Forceman antigens. This lipoteichoic acid consists C polysaccharide covalently linked to lipids. Capsular antigens. Capsular polysaccharide is antigens in human and rabbits. The capsular polysaccharide is also called specific soluble substance, as it diffuses into the cultural medium or infective path or host tissues. Streptococcus pneumonia are classified into more than 19 different serotypes based on the antigenic structure of the capsule polysaccharide. Function: antiphagocytosis capability, inhibition C3b opsonization of the bacterial cells. Protein A, pneumococcal surface protein A, CBPA or PSPA, adhesions and attachment to mucous surface by attaching with N-acetyl galactose. Pili, hair-like structure that extend from the surface, contribute to colonization of upper respiratory tract and increase the formation of large amount of tumor necrotic factors by the immune system during sepsis, raising by possibility of septic shock. And enzymes: neuraminidase, protease, toxins, pneumolysins O, a protein that is cytotoxin to eukaryotic cells that have cholesterol as a component of their cell membranes, particularly the respiratory epithelium. Also activates complement. Autolysin causes lysis of pneumococci in the presence of surface active agent or antimicrobials that inhibit cell wall synthesis. Release toxic protease. On this slide you can see virulence factors of streptococcal pneumonia or factors pathogenicity, factors of adhesion, factors of invasion, aggression, and toxin formation. Pathogenesis. Aspiration of nasopharyngeal secretion into lower airways leads to rapid growth of pneumococcal in alveolar space. Reduce clearance of airway due to disruption of ciliate epithelium in upper respiratory tract by viral infection or smoking promotes aspiration. Antiphagocytic capsule is required for virulence. Pneumolysine, similar to streptolysine or lysis cells. Pneumococcal immunoglobulin A protease cleavers secretory immunoglobulin A and increasing adherence to mucous surface. Neuraminidase spreading factors promotes bacterial spreading into tissue. Pneumococcal disease, typical community-acquired pneumonia, marked by abrupt onset, high fever, rigor, productive cough, and pleuritic change from pleural inflammation from either bronchopneumonia or lobar pneumonia. The epidemiology, reservoir human carriers or sick individual, transmission respiratory auto-inoculation, and temporal pattern winter and early spring. Pneumococci disease, pulmonary infection, upper respiratory tract infection means sinusitis, otitis, cause pneumonia, complication like pleural effusion, emphysema, and can cause extrapulmonary infection, complication of pneumococci disease, meningitis, pericarditis, endocarditis, bacteremia, and another extrapulmonary infection. On this slide you can see clinical features, clinical symptoms, pneumococcal pneumonia, laboratory diagnosis, direct examination of sputum gram strains, cultural method, we use bacteriological technique, study hemolytic properties, sensitivity to optahens, catalase activity, and we use serological diagnosis for detection of pneumococcal antibodies, detection of capsular polysaccharide. For prevention of infectional disease, pneumococci disease, because cause a lot of disease, we use vaccine, 13-valent polysaccharide vaccine, 23-valent polysaccharide vaccine, and 10-valent polysaccharide vaccine. Dose for administrate 2, 4, 6 months. Booster dose at 19-20 months. Main factors which we use for vaccine: capsular polysaccharide. This characteristic of vaccine: Prevnar, Pneumovax, which includes different serotype of Streptococcus pneumonia. Prevnar includes certain serotypes. Pneumovax includes 23 serotypes of Streptococcus pneumonia. On this slide you can see Prevnar, pneumococcal, Pneumovax 23 for specific prevention of pneumococci infection. Thank you for your attention. Don't forget to take the computer test in your personal account.